So we're going to jump right into the talk from John Capitanio. John has been working with monkeys for a long time and has been looking at biobehavioral indices, which is a fancy way of saying personality. So he's been doing a lot of personality work with monkeys and has done a lot of the pioneering work looking at how personality relates to monkey behavior and how it predicts measurements taken during very early life can predict a lot of outcomes later in adulthood. So without further ado, John.
Thank you all, and thanks, Eric, for inviting me to this. I'm very pleased to be here and hope to have some conversations with folks as the day progresses. What I want to do is talk largely about a paper that we just had published recently that has a similar title to the title of my talk, but I'm going to kind of go through it in a fair bit of detail. I find myself at the age of 62 recognizing that males and females are different. And I mean, you know, I've known it since I was about 10 years old in certain ways, but actually there's some really interesting differences, and I'll talk about some of our data that are related to this, but not specifically about pair housing as we go along. So the last set of talks was really a valuable lead up to this, because what that means is I can go through several slides. So first, give you a little bit of an outline of what I want to do. I want to talk about the importance of social housing, regulatory and conceptual issues. In the regulatory part, I'm just going to breeze through. The importance of individual differences, a biobehavioral assessment program that I developed at the Primate Center here in Davis, about 2 miles down the road. And what we found from that program, primarily as it relates to pairing success. And the people that actually did the pairings are sitting over here at this table because it's only 2 miles down the road where they normally work. So I'm not going to spend much time on this. There are certainly very good and very, I think, clear regulatory issues pertaining to the social housing of non-human primates. We've heard about it from USDA. We've heard about it from the guide. We've heard about it from ALAC, and summarized nicely by OLAW, that there is pretty much universal agreement that non-human primates should be socially housed. So what I want to do is just talk a little bit about a couple of other conceptual issues pertaining to this, and then throw a little bit of caveats or cold water or cold tea, as the case may be, onto the discussion a little bit. So rhesus monkeys, which is the species that I study, and are a really cool species, had a divergence with humans about 23 million years ago. So I know it seems like a long time. It's certainly more than last week, but in the grand scheme of things, it's not really that long a time. We don't really know a huge amount about the common ancestor, but we do know that there is a common ancestor that goes back about 30 million years ago called Egyptopithecus. And the fossil data, looking at dentition and looking at bones and those kinds of things, suggests that this common ancestor to humans and rhesus monkeys lived in multi-male, multi-female groups all year round, which is the way rhesus monkeys live. We know that now. But I find this to be really a compelling case for the fact that everything that both rhesus monkeys and humans experience takes place in a social context. So your ability to metabolize glucose properly, anything, your cardiovascular system, your sensory processes, your mental health, all of that has taken place in a social context for at least 30 million years. So to get to a point that Mel made earlier, what's the issue about studying whatever it is you're interested in studying, glucose metabolism or diabetes or things like that, with individually housed animals? Sometimes you need to, but because those health problems for humans evolved in a social context along with us, it suggests that social housing is really the appropriate context for us to study them today. And it presents some challenges, but I think we all are aware of what many of them are. But it's also, I think, from an evolutionary perspective, a really sensible thing to do. So in the last few years, we've been working on a project studying naturally occurring loneliness in rhesus monkeys. So if you look at the social propensities of animals, some animals are really highly social, some animals are not so highly social. And of those not so highly social animals, there are really two types. There are the type that we would call introverted. So these are individuals that can socially interact, but given their druthers, they probably would choose not to. And then there are animals that are low in sociability that really want more than what they have. We consider those to be lonely animals. And so this is not unlike the situation with people. We all know introverted people. They're not social basket cases. They can interact fine, construct full-length sentences, things like that. I consider myself one of them, so I can make fun of us. But I would rather pretty much sit at home and play my guitar or read a book instead of go to a bunch of parties every Saturday night or something like that. Well, what we know about loneliness in humans is it's associated with elevated morbidity and mortality. It does seem to exist in non-human primates. We've identified this in a behavioral paper that came out in 2014. Seems to involve dysregulated immune responses. Paper just came out in December on that, November, December, something like that, looking at what's going on in lymph nodes, what's going on in the context of a pathogen, that sort of thing. There are definitely some internal sort of factors there. And the way we sort of start thinking about loneliness is that we think about loneliness like hunger. Hunger is a signal that we have to tell us that our body needs something, and it needs food. And we think that loneliness is something similar to that, though at a more psychological level. If we're feeling lonely, that may be our 23 million years of evolution telling us we need something. So go out and try to get it. Now, there's a huge literature in humans about loneliness and about some of the psychological aspects associated with loneliness that prevents people from just simply going out and doing it. And a lot of it has to do with perceptions of threat by your social initiations. So it's not just, oh, you're lonely, go join a club or something like that. There's more nuance to that than just simply something like that. So both of these things I just want to sort of summarize by saying the social nature of a social species is really embedded in its basic biology. It has an influence over the course of 23 million years of evolution to have an impact on a lot of physiological as well as behavioral processes that take place. So that's kind of how I think about the need for social housing and socialization and social experience. And we've done a lot of studies over the years looking at stable social conditions and unstable social conditions, which I'm not going to talk about at this point. But I think the bottom line really is that we're social critters, and the social environment is going to have an impact to a greater or lesser extent depending on the physiological or behavioral process that you're talking about. But it's going to have an impact on probably the majority of processes, since we've evolved within that context. Okay, so caveats. So it's important to remember that the reason we have monkeys in captivity in the first place is for the science. Okay, so there's always going to be a need for individual housing. We've heard about that already. We know that there's mechanisms in place for doing that. But I think that it's important to just reiterate that the reason that we have monkeys at places like primate centers and in our labs is because we want to do science with them. Pair housing is not a panacea. John Bowlby, who studied, really kind of defined the field of attachment back in the 1940s, 1950s, infant-mother attachment, developed this concept called the environment of evolutionary adaptedness. So I just told you about 23 million years. So our environment of evolutionary adaptedness includes all of the kinds of things that have sort of been relatively constant, although things come and go over the course of that 23 million years. So I would argue that our environment of evolutionary adaptedness involves much more complexity than what we're going to see in pair housing. Okay? Now, you could scale things if you want and say, well, you know, pair housing is way better than single housing. Got no problem with that. Is it that much less good than housing in trios or housing in tetrads or housing in a large social group or something like that? Nobody really knows the answer to that question. Okay. But just, you know, our environment of evolutionary adaptedness is different than living in a small cage with another single individual. Not every animal in any social group is going to derive the same benefit from it. My favorite example was a monkey that I had in an SIV study a long time ago, who we studied in our social field cages that many of you saw on the tour here yesterday. His name was Phaedrus, great guy. In his field cage, he was skinny and bald as a cue ball and just kind of running back, deer in the headlights look in his eyes. I mean, there's just really no question about it. And here he was living, you know, in California, Northern California. You know, he never got the wine or anything, but, you know, sunlight, a lot of great environment. We recruited him into a study for looking at the effects of social stress on SIV disease progression. We brought him indoors and we housed him individually. And over the course of a few months, gained weight, grew this magnificent coat of fur, and generally seemed to calm down a lot. So I would argue that by some indicators of welfare, he was probably better off sitting in a room in an individual cage than he was in that specific cage. Now, that cage could have been changed so that his welfare might have improved in that environment. But just as an anecdote, that has stayed with me for about 23 years since we actually did that study. So we don't usually think about things like this. But what we're doing with our animals when we do science is we are developing a model of something that we want to know about in another species like humans. So an animal model of diabetes, or an animal model of cancer, or an animal model of something. Okay? We know that a model is not the same thing as the thing that's being modeled. You can learn a lot about a model of a bridge that's made out of balsa wood, but when I run my truck over it, you know, what comes out the other end is just simply going to be a bunch of kindling. Well, that idea actually applies to what we're actually doing in terms of behavior management. It's not only true of the science, but it's true of our captive care programs. We are modeling the evolutionary environment of these animals. So forage boards model foraging that animals have been doing for, you know, 23 million years. Pair housing models the social complexity. And I'll just simply go back to what I said about models when I was talking about science. The model is not the same thing as the real thing, because if it was, it would be that thing. It wouldn't be a model. So this is the struggle that we have in terms of captive care, I think, too, in that we are trying to extract some of the essential features out of that environment of evolutionary adaptedness to provide the best environment that we can as a compromise between, you know, the monkey living in the perfect set of circumstances and the monkey being in our studies. So you would say, well, that seems to create a problem, because how are things going to generalize if you're looking at this really abstracted version of reality that the monkeys are in? And I would argue that generalizability is always, always, always an issue. I would even put a fourth always in there as an issue. And it's probably one of the things that scientists pay the least amount of attention to. Okay? So a lot of psychological research is done on college undergraduates. Okay? They're good models for something. And, you know, I'm willing to bet that their action potentials work exactly the same way as my action potentials, for the most part. But, you know, not a lot of life experience with that subset of individuals, and so if you're looking at things pertaining to social relationships, maybe or particularly heterosexual relationships, there might be some question marks about the generalizability of your results. Generalizability, that is the results that you gather from your own research project, generalizing to the broader universe of individuals, people, other monkeys, whatever you want to call it, is something that we always need to be careful about. Okay, let me talk a little bit about individual differences. So individuals differ from each other in a lot of different ways. Some of us are tall, some of us are short. Some of us drink too much beer and carry it around in front of them. Proudly, I might add. The origins of these individual differences come from a lot of different places. Our interest is in behavior, and that pretty much means the observable actions of the organism. And what is behavior all about? Behavior is the mechanism by which individuals get their needs and wants met. So if I'm a tree, I can just sort of stand there and expect that a lot of what I need is going to come to me. Okay? But if you're an animal, you are probably going to have to go and get it. So what our behavior does for us is it gets us what we want, given the things that are around us in the environment, so the opportunities that the environment presents. So I said that behavior, this mechanism that mediates the needs and the wants, comes from a lot of places, but one place it comes from is temperament. Okay? And temperament, which in the animal world we usually use synonymously with personality, that's actually becoming a lot more common in the human research world too, not the human world, we're in the human world. But Gordon Allport, back in 1937, very prominent personality psychologist, said personality is something and does something. It's not synonymous with behavior or activity. It's what lies behind specific acts and within the individual. Okay? So personality or temperament is what we bring with us to the situations that we encounter all the time. Now, how that personality gets manifested in behavior depends on a lot of different things, largely about the context that we are in and what the opportunities are out there in the world. But I just simply want to make the distinction between behavior and personality or temperament. So, what are there for temperament traits? This is a listing of temperament traits that most human researchers would agree with. There's not a lot of them. Behavioral inhibition, activity, some animals are much more active than others. People, not animals, these are human temperament traits. But you can probably see that they have their analogs in the behavior of the animals that we're working with. Empathy, agreeableness, maybe not so much when it comes to non-human animals, at least in the same way that they're talking about there. So there's a finite number of temperament traits, and they're all about these kinds of things: activity, emotionality, the ability to inhibit our behavior when we need to. Okay? And a lot of what I've been working on for the last 15 years in this biobehavioral assessment program is try to understand the causes of a lot of the variation in these factors, as well as the consequences of it. So let me explain to you a little bit about the biobehavioral assessment program here. So it's a program that we developed a while ago, the year 2000, 2001. It is a colony-wide 25-hour assessment of infants. The infants are 3 to 4 months of age. We take them from wherever they are in our colony, and we house them individually for this 25-hour period. We run them through a series of behavioral and physiological assessments pertaining to physiological reactivity, emotionality, temperament, so forth. And the goal is to follow these animals and see what becomes of them. Are things that we're able to see when the animals are very young persisting into adulthood or at least into older age, and how does it map on to health consequences, for example? So we make this information available to colony management people, folks over here use it. We make it available to researchers who want to select animals for their studies, and there are whole 3 R's kinds of things about that. You can select a more homogeneous sample. If that's what you do, your variation is going to be lower. That means you need fewer animals for your study. However, your results may not be generalizable to the world at large. It's only generalizable to the kinds of individuals that have those characteristics. But we don't need to go into that at this point. The animals in our program come from all 4 quote-unquote colonies at the primate center. Field cages, corn cribs, which are these smaller social units at the primate center. Nurseries, there were many more nursery-reared animals in the past. That was the manner that was used to derive specific pathogen-free monkeys at our colony. The nurseries are much more reduced than they were. And animals that are indoor housed. So oftentimes these are animals that are part of our time mate program. A female that's typically paired with another female and a 4-pack like this. And then they're brought to the cage of a male periodically, and males and females do their thing. And so we know pretty well when conception takes place, and that can be very useful for particular kinds of studies. So over the course of this 25 hours, we assess behavioral responses to separation and relocation, HPA regulation. We do like a dexamethasone suppression test because we want to look at the integrity of the HPA axis. Quite variable, actually. We do a CBC on the animals. We want to know who's got a lot of CD4 cells, who's got fewer CD4 cells. We look at a genotype for 2 promoter genes of neuropsychiatric interest to us, serotonin transporter and monoamine oxidase A. Test of visual recognition memory, responsiveness to videotape social stimuli, responsiveness to a human challenge. This is a human intruder test, which is something of a staple in non-human primate laboratory work on temperament. We give them a novel object that's got one of these actimeters in it so we can assess how much they interact with the novel object without having to stand there in front of the cage for 25 hours and record that behavior. And then we do overall assessments of temperament. And the measures that we primarily work with are these measures of, first of all, behavioral responsiveness. So within about 15 minutes of them arriving in our luxurious testing suite, we do 5-minute focal observations on these animals, and we've been able to do exploratory and confirmatory factor analyses and find that we can explain the variation in these data by 2 dimensions, activity and emotionality. And we assess that on day 1, and then we assess that just before they go home the next morning to their moms. The human intruder tests that I talk about, there are, again, a factor analysis, confirmatory and exploratory factor analysis shows that there are 4 factors. And then temperament, which we assess at the end using a rating methodology. Now, a lot of what I'll just simply point out here is that these are a lot of words and names, but a lot of what we're talking about refer to things that are basic indicators of activity, basic indicators of emotionality. That would be akin to the activity and behavioral inhibition and aggressiveness and irritability that I showed on the human temperament slide before. So we're sort of tapping into similar kinds of constructs, although we call them by different names for specific reasons. So, okay, great. We're assessing these things when the animals are infants, 3 to 4 months of age. How stable are they across time? Well, they're pretty stable across time. So infants that were behaviorally inhibited, that is, they tended to stay at the back of the cage and just kind of keep an eye out, just watching what's going on, not jumping around, not doing a lot of vocalizing. If you test them years later, they're much less likely to accept a food treat, and they're much less likely to display emotional behavior when you take them from their cage and put them in a single cage for a brief period of time. Okay? So this element of inhibition seems to be constant. These inhibited females are much less likely to take a food treat if you just walk up to the cage and put a grape out there. Okay? If we do attitude ratings when the animals are adults, we can see that there's 73% agreement in the rating of nervous when an animal is an adult compared to the rating of when the animal was 3.5 months of age. So this is spanning, a 5 to 10 year age range. We've got a bunch of other data. So there are measures of this infant assessment program that map onto things like the age that females first reproduce, the number of offspring adult males have. when they sire offspring in a field cage. The occurrence or the frequency of diarrhea. So we've looked at a lot of outcome measures, and what is remarkable, although not terribly surprising, knowing the human literature, that very early indicators of temperament stay with individuals for their lives. It doesn't mean it's a destiny, doesn't mean it can't change. Okay? One thing that I didn't put up here, but I'll just simply mention now, we were very concerned when we first started this program that there would be, hopefully and presumably, no long-term consequences of this brief 25-hour separation that these animals undergo. And we knew that if animals are at an older age and they undergo a longer period of separation, that there are lasting consequences. And the way that we know that is through 3 publications that I published back in the 1980s. So I kind of had a leg up on what to look for. And we did some really extensive testing up through a couple of years of age on these animals. And everything came out to be not significant. So we think that our experience with these animals sort of taps more into the human literature pertaining to resilience than it does to long-term adverse consequences of this. And now with open access papers, we're going to try to publish it. It's really hard to publish a paper where P is never less than 0.05. Journals just don't like that kind of stuff, but with open access journals, we can do that. Okay, so I want to talk about pairing. Here, behavior, temperament, and physical variables as indicators of pairing success. And this is coming from a paper that was just published in the American Journal of Primatology. It's part of a special issue in AJP, edited by Kate Baker and Amanda Detmer, titled The Well-Being of Laboratory Non-Human Primates. If your institution doesn't have a subscription to this, send me an email. I'll send it to you. And hopefully no representative from Wiley List is going to be watching this video and start having a problem with my doing that. But I'll do it anyway. So. Obviously Brenda McCowan is a co-author on this as well. So let me first just say, other people have kind of alluded to this, but I will just make it pretty blunt. Individual characteristics are properties of an individual. Pairing success are properties of a pair. And so these two phenomena are at 2 different levels of analysis. General systems theory tells us that it's real hard to predict something that's at an upper level of analysis by knowing things that are at a lower level of analysis. And the reason for that is things at an upper level of analysis usually result in emergent phenomena occurring. So I would argue that pairing success is an emergent phenomenon of the interaction of 2 individuals. Success doesn't inhere in a specific individual A or individual B. Success only is present or absent based upon how the 2 individuals interact. Okay? So dominance is another good concept. There aren't dominant individuals. I know many of you believe that there are, but really there aren't. Dominance is actually a characteristic of a relationship between 2 individuals. You could have a confident individual. You can have an individual that shows surgency. But in the case of using the word dominance in the way of describing a relationship, there aren't dominant individuals and subordinate individuals. There are timid individuals and there are confident individuals. Okay? So, pet peeve. So our goal was to try to create, dyadic measures so that we could try to get at success in a little bit better way than just using individual level measures. And I will talk to you a little bit about what those measures are. So what we did was we mined the database that these folks at this table have been creating over the course of, I don't know how many decades of pairing attempts. Animal A, animal B, we put them together on this date. This is what happened, here's the outcome. So we had 340 isosexual pairings, about equally split between males and females. A total of 493 animals, so obviously some animals appeared more than once in our data set. All the pairings occurred between 2007 and 2012 that involved both members that had been through our BBA program. They came from everywhere: field cages, corn cribs, indoor nursery, the indoor mother-reared colony, that sort of thing. And they ranged in age from 1.2 to 11.1 years. Okay. And the methodology that our pairing staff uses is they move the pairs to an adjacent cage with a sliding partition. They open it up a little bit. If the animals look okay, they'll open it up completely. If the animals look nervous, they may put a grade in for a period of time. And the pairs are monitored for a variable period of time. And success is defined as animals showing affiliative behavior. Now, not everybody uses the same definition, but that's the definition that's used here. They're consistent dominance and subordinance roles, and the animals don't appear fearful of each other, except maybe during feeding time, which may be somewhat understandable. Of our 340 pairings, 121 were unsuccessful, 219 were successful, and yes, there was a sex difference. A little over half of the male pairings were successful, but almost 3/4 of the female pairings were successful. So the analysis that we looked at was just simply a simple outcome measure. Did these folks judge the pair to be successful or not, 0 or 1? Actually, it would be 1 or 0, but you get the meaning. We had a suspicion that something might be going on differently between males and females, so we ran separate analyses for males, pairs, and separate analyses for female pairs. And we put in these covariates, which was not what we were really interested in. I mean, we were interested in them, but I really wanted to look at these temperament measures and see how that mapped onto it. So that was kind of the main orientation here. Figured it out. So the ones that are in red are difference scores. So these are measures that we think are getting at something pertaining to the dyad. So did the animals have the same or different rearing? Now, I showed you that we had 4 rearing conditions, but actually we collapsed it because it was going to make our head explode if we tried to code for a field cage reared animal paired with an indoor mother reared animal. There were just too many combinations, and we would finally get down to really super small sample sizes. So we just simply looked at indoor versus outdoor. Okay? So were both members of a pair from indoors versus were they from outdoors? Or were they were they mixed? The weight difference at time of pairing. So what did they weigh? We just subtracted one number from the other number. And the age difference at the time of pairing, same thing. Whoops. So we ran 4 sets of analyses, and for each set of analysis, remember I showed you 3 sets of data that are our principal measures. So we ran our covariates with the behavioral measures, activity and emotionality. We ran our covariates with the human intruder measures, activity, emotionality, aggression, and displacement. And we ran our covariates with the temperament measures, nervous, vigilant, confident, gentle. And then when we found significant results from the behavior and temperament measures, we ran a final analysis throwing them all in to see if they still retain their predictive ability. For the various BBA measures, nervous temperament, for example, we calculated 2 scores for the analyses. So I have a score for nervous temperament. Eric has a score for nervous temperament. So we can calculate 2 indices of our nervousness, okay? One is a mean score, so Eric's score is 7, my score is 3, sorry. So our mean score would be 5, okay? And we can also calculate a difference score, so how discrepant are we? So our difference would be 4, okay? So those were the measures that we looked in because we wanted to kind of look at more dyadic level measures. Okay, well, what did we find? We found something really pretty interesting. So for females, we have all of these predictors of pairing success, but everything is non-significant except for those 3 measures. Those 3 measures are the day 1 emotionality score difference. So this is how much emotionality these animals were displaying, 15 to 20 minutes after they came over to our luxurious testing suite. Okay? And it's the difference score. It's not the mean. Okay? Nervousness, another emotion-laden score, and the human intruder test emotionality score. Okay, so what do we have here? What this is telling us is that predictors of, and all of these covariates were not significant. What this is telling us is that for females, individuals that were more similar to each other were more likely to be successful. Okay? So if you have a highly emotional female, if she's paired with a highly emotional female, you are much more likely to have a significant, a successful pairing. Okay? So for females, phenotype matching seems to be the order of the day. Now, any of you who are at primate facilities, you don't need to employ a biobehavioral assessment program at 3 to 4 months of age. You can simply assess emotionality of your animals and get a handle on this without going through, you know, in another talk I've talked about the BBA program as a Cadillac program. Okay? You can develop a Kia program. Okay? That will get you some of the same answers. Okay? We've done the heavy lifting here with the BBA program, and here's what we want to pay attention to when it comes to females. Okay, what about males? Well, males are a problem. Things are very different for males, and let me take you through this one at a time. Okay? So the first thing is outdoor rearing. Success among male pairs was facilitated if both members of the pair were reared indoors. Okay, well, why is that? Well, we suspect it's because, you know, pairing takes place indoors, and animals that were reared indoors are much more familiar with the routines of morning health check when somebody comes over and stares at you and, you know, looks at your legs and your butt and things like that. They're more familiar with the whole idea of hosing. They're more familiar with seeing lots of people and that sort of thing. So indoor reared animals were more likely to be successful, although there's going to be a caveat to that in a few minutes. Pairing age. So if you're younger, at the time of being paired, you are more likely to be successful. People that pair monkeys are probably not terribly surprised at that. Okay. Differences in weight. So the larger the difference in weight, the more likely you are to have a successful pairing. We've known about this in my lab for a while from early studies that we've done 25 years ago on SIV, looking at monkeys in stable versus unstable social groups. Disease progressed more rapidly in monkeys in unstable groups, but because our social groups were quite small, we ran into a situation where 2 of the 3 animals, adult males in this SIV study, would be euthanized. And so we wanted the study to continue, so what did we do? We went back to the early literature that was done with the Wisconsin isolate-reared monkeys, looking at how to rehabilitate them. And what they said in the late 1960s, Steve Sumi and others, is that if you take these, you know, larger adult animals that were isolation reared, first of all, if you pair them with a novel adult animal, all hell is going to break loose. But if you use really younger monkeys, which they refer to as therapist monkeys, you're more likely to have successful pairings. So we did that with our SIV-infected monkeys, with the remainders of the study of monkeys in socially stable conditions, and it worked. Okay? So this seems to indicate that as well. So differences in weight, not necessarily differences in age. Okay. Differences in rearing. So if you had similar rearing, you were more likely to be successful as well. So indoor with indoor. Again, we're not differentiating here between nursery and indoor mother reared. Outdoor with outdoor. What's that about? Well, if you can imagine, you know, a monkey that's coming from the outdoors that is paired with a monkey that's from indoors, the monkey coming from outdoors has got to deal with a lot of stuff that the monkey that's been indoors for a long time doesn't have to deal with. It's like, who are these people coming around squirting jets of water in my cage every day? You know, feeding is done differently. The light-dark cycle is done differently, that sort of thing. So with individuals sort of matched for their degree of experience, either indoors or outdoors, you're more likely to kind of be dealing with each other and not dealing with each other, plus the unfamiliarity with hosing and cage change and all that sort of thing. So here's that interaction I was telling you about. So if you're an outdoor animal, the later that you are paired, the better. We've also got some other data from a study of motor stereotypy that the later you're brought inside, the more kind of protective that is. Okay? And so, you know, there's a thing in the literature about if you're lucky to have cages like we have out here, keep the animals out there for as long as you can, because there's an incremental benefit to every day or week or month, whatever unit you want to talk about, that they're in that environment. When you bring them in eventually to participate in your studies, the longer they've been out there, the better they're probably going to be able to adapt to the situation. Well, that was all very interesting, but we were really interested in the temperament stuff. And so here it is. So if you have lower scores on these temperament traits of gentleness and nervousness, you're more likely to be successful. And this is after controlling for all of those other factors that I just talked about here. So what's important to note is that it's the mean values that are here. So if you have a high nervous male, you're probably going to be more successful, all of these other things being equal, if you put that animal with a low nervous male, because it's the mean that seems to be predictive of success. If you have only 2 high nervous animals, you may not be able to pair them, okay, because their mean values will be high. Okay? So nervousness is a temperament characteristic having to do, very similar to the human temperament characteristic of neuroticism, major personality factor, and we've done several studies looking at animals with a nervous temperament. They show evidence of glucocorticoid insensitivity. Like individuals that are high in neuroticism, they tend to see the glass as half empty. We don't actually test them with a glass, but you know, if you give them a standardized situation, they're much more likely to respond not with anxiety, but with aggression. They're the ones that are shaking the cage, okay? They're not scratching themselves and yawning, shaking the cage like that. So nervousness as a temperament factor is a pretty significant factor, and it's cropped up in a lot of our different outcome measures. Gentleness seems to refer more to, we think, with having an active coping style. So if you're low on gentleness, we think, you are more likely to actively try to engage the environment. So we find low gentleness actually correlating with motor stereotypy. So motor stereotypy used to be thought of as bad, 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 and now it's not quite thought of in that same sort of way. There's evidence to suggest it might actually be an adaptive response in an adverse environment. So animals that are low in gentleness are much more likely to be more active in terms of pacing and trying to manage the situation that they're in. Okay, so I would argue that behavior and temperament can be useful in facilitating successful pairings in captivity. Again, you don't have to implement a Cadillac version of this. You can implement a Kia version of this. Okay? Factors that are influential differ for males and females. So for females, phenotype matching seems to facilitate success. And those other covariate things didn't really seem to matter at all. For males, the other covariate things were pretty influential. And in fact, the different scores that we saw in terms of weight were really an important factor on this. What it suggests is that at least in terms of temperament, there may be some males that are going to be really hard to pair. Okay? So if you have animals that are high in temperament, and you happen to be at an institution that only has 14, adult male macaques for neurophysiology studies, you may be out of luck in trying to find a compatible partner for your animal. But the important factor is it's the difference or similarity for females and for males. It's the mean value. So I haven't said why. Sex differences are related in many ways, including in our own species, to our life history characteristics. So in rhesus monkeys, females are born into a group. They're going to stay in that group the whole time. When they're in that group, they're going to be interacting primarily with kin. The females that they're going to be interacting with in that group are likely to be similar to themselves because they not only share genes, since they're related to each other, they're kin, but they also shared environment. You know, you're a newborn female in a rhesus monkey group, you know, there's your mom, there's your aunt, there's your sister. They're all going to be there for the next 20 years, unless, you know, they're harvested for projects or whatever's going to happen. But think about it in the wild. They're going to be there forever, okay? Leopards notwithstanding. Males are really different, okay? So the point about females is the fact that females that show similar kinds of patterns of emotionality may not necessarily be surprising, because in the wild, that's who they interact with, kin, who are going to be similar to themselves in terms of their pattern of emotionality because of shared genes and shared environment, okay? Males are really different, okay? So males are all about rank and power, as we heard from Brenda's presentation earlier. So they'll compete for rank. We know that that's the case because males are larger than females. And in species where there's sexual dimorphism, that usually has to do with members of the larger sex competing with each other for access to members of the smaller sex, in this case, males and females. So things like weight, you know, may not be terribly surprising. Interestingly, some environmental variables that we saw were important for males that were not important at all for females, weren't even on the radar screen for females, such as similarity in rearing, or, you know, being reared indoors and so forth. We've actually done some heritability studies of many of these behavioral and temperament measures that I talk about. Turns out for some of these measures, the heritabilities for females are significantly higher than the heritabilities for males. So what that means is, turning it around, is that for some of these measures, males are just more sensitive to the environment than females are, okay? And if you think about what males have to do in their life history, they got to leave their group, they got to find another group to get into, or they got to find, you know, some other males to kind of hang out with for a while. Then they have to finagle their way into group. They have to read the situation. They have to, you know, they have to adapt in many ways in terms of patterns, particularly of emotionality. That's not something that females really necessarily have to do, because they stay in the group that they're born in all the time. So, you know, in terms of temperament, there are other issues that we could talk about here, but the point that I simply want to make is, if you look at any species, it's got an evolutionary history. And depending upon what the life history variables are pertaining to males and females, this idea that I'm presenting here, that there are going to be sex differences in what's important, become a little bit more understandable. It's not just a random thing. These animals have an evolutionary history, and the agendas of males and females, just like here, are often different. So I want to acknowledge collaborators here, the technicians in particular, the veterinary and animal care staff. A lot of the folks that are sitting at that table collected some of the data that went into this, and that's it. So thanks.